Welcome to our CalCOFI lab aboard the Sally Ride. Here we are filtering for eDNA using the MOSAIC protocol. MOSAIC stands for Murray Ocean Sampling Across an Integrated Continuum. And Murray is a project where we're looking for marine mammals in vast swaths of the ocean. So we're gathering really large volumes of water in these large carboys and using these Swinix housings, like so, we are able to gather up to 50 liters in these 5-micron filters. So I'm about to show you our exact method for how we accomplish this. Okay, so now we're going to go collect some CTD water. This is a fresh whirl pack with uncontaminated so far, so we're going to open it up for this sample. First, we get on our safety gear.
This is important information. Cast 41.
Okay. Whenever we go out to deal with the CTD, we wear a life vest and a hard hat. Safety. So now, generally the bottle that we take from is the one that we're going to use to compare to the surface water samples. So we take from the surface CTD bottle.
Shall we check the laptop for which is the surface CTD bottle?
Yes. So we're going to come around here, check the laptop. Usually it is the highest number that has a lid on it, but we're going to be sure and we're going to check all our info here. So it looks like that would be bottle 16 at 2.9 meters. This is from the last cast, but we're just going to demonstrate.
This is the time the CTD came up, the coordinates you should use for its location, and process number 40 means it was cast 40. You should record cast, bottle number, and depth and coordinates for all CTD samples you take in your lab notebook.
So then, this nozzle will generally have a good flow rate, unless a lot of people have been using the CTD water, then this will be out and you can use this lower one. I generally use the lower one just because we're trying to get a whole bunch of volume. So you operate these by aligning the pin with a little hole in the handle here and press it in. Now if this were full of water, this would be spewing out very quickly, ideally into the bag that we have prepared.
Stick the bag right up against the nozzle and don't forget your rubber boots.
Yes. Which I am not wearing.
Me neither. That's okay though.
Then, after our bleaching and cleansing is done, we would immediately swap out this bag, put it in this bin, and place our water in here and begin sampling. So in order to compare our MOSAIC eDNA sampling method with more traditional bottle sampling type methods, like from the CTD rosette of Niskin bottles that we'll show you in a minute, hopefully, we are filtering out of our CTD water, which we collect in these sterilized Ziploc bags, and filter with these serological pipettes, which we have removed the cotton filter from. Very important to remove the cotton filter. And we have the same situation going here, where after it passes through this peristaltic pump, it comes out into this filter, same type of filter that we're using for the underway samples. Here. and passes into this bag where we just measure the volume that has been filtered already. But before you measure your volumes, after you finish sampling, it's important to get that filter stored and processed as quickly as possible.
It's important to unclip the peristaltic pump so that it doesn't chew up your tubes.
Yes. So now I'm going to get a microcentrifuge tube labeled for our CTD water, which is very important to keep good notes. This is going to be filter 164 of this cruise. And then we work in this portable hood, which is very exciting. You open the door, it already has its blower going and has the ability to UV your housings after you clean them. So before I open this filter, I'm going to gather my supplies, make sure everything's ready, and open it up. Take note of whether there's any bubble noticeable. That would be a significantly lighter spot. And I don't see any, although this filter is very light. So then we're going to grasp it with the forceps and fold it in half. Fold it in half again. And then you can turn your forceps to kind of press it down to hold the fold in, like so. And I'm gonna slide it right off the edge there. And now it's ready, now that it's folded in eighths, to place in the tube. Now, these tweezers are done, so I'm going to put them in our pile of tweezers to clean. And get our Zymo DNA RNA shield. You can use any preservative buffer like this, but we really like DNA RNA shield. And we're using 400 microliters to preserve it. This protects against freeze-thaw cycles and also against degradation of your DNA. If you don't have a freezer aboard your research vessel that you can use, it will preserve your DNA at room temperature, but our protocol is to then put it in a negative 80-degree freezer. Boosh! Yes. And place it in there. Ready to transport home to the lab to extract the DNA from. So now, after we have stored our CTD sample, I'm going to measure how much water flowed through that sample. next sample, number 164. Okay. And our end time, we have to note our end time, is 12:32. So now that we have completed filtering a CTD sample, we're going to bleach the hose and DI water the hose in preparation for our next sample. So the first thing I'm going to do is run the remaining sample out of the pipette. So I'm going to clamp down its tubing and run its controller on reverse. Okay, there's actually none left in there, but always good practice just to make sure. Turn that off. And now we can get out our bleach bag. Now we are reusing the bleach. We're not allowed to put any bleach down the sink. Yes. So we will be running the bleach into and out of the same bag. So we grab the end associated with this tubing right here, and we're going to put it so that it flows back into the bleach bag, like so. So my pipette's in there, outflow end, and I'm just going to close it down so we don't get any spillage. Now, I'm going to run it on forward, and we'll start pumping at a relatively fast speed here just to get the air bubbles out. Yep, 8 or 9 is good for getting the air bubbles out. You can hear the tapping of the air bubbles passing through the hose. So that will help us suck up bleach. And then we should start to see it coming out the end here.
There it is.
There we go. And so now we can reduce this speed to a 4, set a timer for 10 minutes, and that will pump the bleach through for 10 minutes. After we're done with that, we're going to swap bags to this DI water bag. Same thing, blow the bleach out in reverse, put this in here, but we're going to then take the outflow tubing into an outflow bag for DI water that we're then going to dump, so that our DI water remains uncontaminated with bleach or sample. And that's it.